Well, thank you everyone for joining. I'm delighted that we have so many people registered and joining. We actually had 900 participants registered for this event. We normally don't get that many actually arriving, but judging by the speed of people joining, it looks like this is going to be a very well attended event and focusing on rare diseases in both in pediatrics and cancer in pediatrics. So it's clearly a compelling and exciting event. So to introduce myself, my name is Parker Moss. I'm the Chief Commercial and Chief Partnership Officer at Genomics England. This is actually the first research seminar that I've hosted, so thank you all for the welcome. And it's also the first research seminar that we've invited a commercial partner to speak, and I'm very excited soon that we'll introduce Velo Health and Yeting to present on behalf of this fascinating company in New Jersey. But before I introduce Velo, I'll just go through a few housekeeping items. So one of the reasons we've invited Velo Health onto the call today is that we're very much trying to integrate the academic and the industry research partnerships and drive collaboration between these two communities, which I think are getting increasingly interested in each other. And indeed, in a discussion today with Yeting, Velo Health actually do have a collaboration mechanism and are extremely interested in reaching out and working with clinical academics in both neurodevelopmental disorders, neurodegeneration, and cancer and cardiac disease. So I think that's something which we can discuss later on in the call. One of the things we're trying to achieve at Genomics England is to really embed participant data far more deeply into the drug discovery process. And what we're trying to achieve is a kind of a concept of a clinical return on data. So what does that mean? We're trying to generate a return for our participants, thanks to their kind of kind and generous contribution of their tissue and their data and the lifetime follow-up that we work with participants on. But we're also trying to increase the return on data for the UK PLC. We want to attract more R&D investment and more clinical trial participation in the UK. And Velo Health are exactly the kind of company we want to work with in this respect. So Velo is a truly kind of end-to-end target discovery and clinical development platform. They use human data and machine learning throughout the process. And we're really pleased that they've begun to make use of gel data, and we wanted them to kind of showcase their efforts in this seminar series today. So just to go through a few points on how this seminar works. First of all, as I think you can all see, this seminar is being recorded, and we make all our recordings available on our YouTube channel within a day or two of the session finishing. So please check that out in a couple of days, and also check out some of the really, really interesting previous talks that are available up there. We do run a Q&A at the end of each session. And I think you'll see that there's both a chat and also a Q&A section. Please make sure you put your questions in the Q&A part, not in the chat part. I'll be reviewing those at the end. If I understand the questions, I'll ask them myself. If you put a particularly detailed and complex question, I may ask you to kind of put your microphone on and ask the question directly. So why are we doing this? Well, what we're trying to do is really to encourage researchers to present and to showcase their research. So please, if you have a research paper that you're excited to showcase and get exposure to, please reach out to our team, and we'll make sure that you get registered for our seminar series. There's very high demand for this. It's booked up months and months in advance, so please anticipate that and reach out to our team, and would love to work with you on that. I'm going to just showcase the next month's seminar. If you could go to that slide, Sam. Yes, we have Dr. Ruth Horn coming in. She's going to be presenting on the ethics of genomic medicine, redefining the values and norms in the UK and in France. So that would be interesting to have a transnational perspective there. And Dr. Sisodia, he'll be looking at the power of the 100,000 genomes for the benefit of people with epilepsy, which may overlap with some of the talks today as well. Very importantly, we ask for a seminar survey to be completed, and we pay very close attention to that. So sometime during the survey today, during the seminar today, Sam will be posting a link to the survey in the chat, and I'll be really grateful if we could have a kind of high level of compliance to that, please. We look carefully to see your feedback and to make sure that influences our future seminar choices and the kind of structure as well. And lastly, we're active digitally and on social media. One thing you can do is you can look at our research portal, which shows all of the ongoing research projects, of which there are very many. So that link will be available soon as well. We have an active Twitter channel, so check us out on Twitter. I've already mentioned our seminar videos. And lastly, we have an increasingly popular G-Word podcast by our CEO, who has got some very high profile guests who come on and speak to us on a monthly basis. So do check out the G-Word as well. And last thing to say before I introduce the guests, the research seminar next month is an important one for us because Mark Caulfield will be hosting that one. Mark Caulfield, our chief scientist, who was our founding chief scientist and has really been here from the start at Genomics England, is leaving to go on to green pastures. You won't be hearing the last of Mark. He has some very interesting things to go on to do, but he'll be hosting the last research seminar in a month from now. So please come on and join not just those great speakers, but Mark's farewell seminar as well. So let me introduce our speakers today. We have Dr. Yeting Zhang, and she has a remarkable background. Yeting is a data scientist working for Velo Health, as I've mentioned. She pursued her PhD at Pennsylvania State University, majoring in genetics and focusing on next generation sequencing data analysis. Afterwards, Yeting finished her postdoc training at Rutgers, working on whole genome and whole exome sequencing to understand the genetic components behind neurodevelopmental disease. Yeting is passionate about leveraging big data and computational capability. She went to the pharmaceutical industry as a computational biologist after her academic time. One area that of our current focus is machine learning approaches in omics, and she's going to elucidate the complex disease etiology that Velo used to generate hypotheses and insights in drug discovery and drug development, which, as I mentioned, is a deeply integrated end-to-end process at Velo. And although Velo work on multiple therapeutic areas today, Yeting's talk is going to be focusing on Parkinson's disease. With that, let me hand over to Yeting. Thank you very much for joining us.
Thank you, Parker. Let me share my screen.
Thank you.
Can you see my screen?
Yep.
Okay, thank you. Go present. All right. Thank you, everyone. Thanks, Parker, for your very kind introduction of both Velo Health and myself. My name is Yeting Zhang. I'm a data scientist working at Velo Health. So the title of the talk is Patient Subgroup Discovery and Insights Generation through a Multi-omics Integration Framework. So before I actually get into any of the details of today's talk, I want to introduce a little bit of the high-level background, set the stage of our interesting project here. So what's the opportunity and the challenges we are facing? So it's almost no doubt that we know that data is no longer a bottleneck to our research. We have ways and access to all kinds of data modalities. This includes real-world evidence data or clinical trial type of data, and also biological level of data. What I mean is saying molecular level of data, which within Genomics England, one of the most advantage is this high quality, high coverage whole genome sequencing data representing here in the green angle here, genomics data. So the opportunity is we have access to all kinds of data modalities, but the challenge also comes along with that is how can we use machine learning approach? How can we integrate all those data modalities? And then through the integration, we can generate insights. And along that process, we can have actionable plan in terms of looking for disease etiology or looking for new therapeutics treating certain disease. And then as myself, who comes from this genetic background, we sometimes look at genomics data, not thinking of it as just the one layer of biological data. If you tease apart the genomics data, you actually can easily tease it into different levels. For example, the common genetic variant can be one layer of the data. For example, a lot of the GWAS study have been focusing on the common genetic variants. And also on the other spectrum, the rare genetic variant, loss, for example, loss of function type of genetic variant, that's another type of the genomic layer. And then coming up to larger scale, for example, copy number variation or structural variation, those are all the different layers. And then, so to introduce a little bit more about our value health vision, for one thing, we definitely want to utilize is the public available GWAS study. And then coupled on top of it is this high quality, deep coverage of whole genome sequencing data provided by Genomics England here. What we want to do and propose to do is project individuals' genetic predisposition to different traits, including both disease risk traits and also common phenotypes. For example, endophenotypes. You can imagine, for example, people who tend to have high level of red blood cell counts or low level of lymphocyte counts, those type of phenotypes. So what we propose is to use Valo's proprietary high dimensional machine learning approaches and stratification approaches to derive deeper insights into both genetic and clinical aspects of patient journeys. So the current stage of us is we have multiple research plans submitted to Genomics England and approved, covering neurological disease areas, cardiovascular diseases, and also partial oncology spaces in Genomics England. So a little bit about roadmap of today's talk. Although we have quite a bit different focus going on at the moment, what we want to talk about today is to pick Parkinson disease project that we have done last year within Genomics England, and introduce more about the computational framework and how we think about how we can utilize different data modalities within Genomics England and solve certain questions in our mind. So introduce about Parkinson disease and also introduce the high level goal. And then also talk us a little bit about the background of two types of genetic aspect. One type is what I already referred to as the common genetic variants. We will use polygenic risk score framework to study it. And then on the other spectrum, the rare genetic variants, we will study the predicted loss of function aspect. And then switch a little bit to talk about our computational framework. We have chosen the multi-omics approach in general, and then come down to show the audience a little some of the analysis results we have done in terms of patient subgroup discovery within PD cohort. This represents about 900 Parkinson disease patients at the time when we conducted this research. And then some of our thoughts about what's next. So coming down to Parkinson's disease, so PD is second to Alzheimer's disease as the most common age-related complex idiopathic neurological disorder. So it's characterized by tremor and bradykinesia and muscle rigidity, along with impaired gait and posture. An estimated 6.1 million individuals globally had a PD diagnosis in 2016. This is 2.4 times higher than the 1990s. So what's known as the most commonly used treatments for Parkinson's disease are only partially or transiently effective and only available or applicable to a minority of the patients. And this is actually one of the main reasons we Value Health think we should do something about this, because we need to find new effective therapeutics for the patients. And in order to do that, we need to understand the disease etiology in a deeper level. And some of the hallmark pathology of Parkinson's disease is this dopaminergic neuronal loss and also depletion of dopamine levels in the striatum. So on this slide, what I want to show is how heterogeneous Parkinson's disease is. So on the top level, top panel, this shows the symptoms of Parkinson's disease. You can see there are a range of different types of symptoms, ranging from early symptoms, motor type symptoms, all the way to non-motor. And within motor, there are also primary and secondary motor symptom difference. Also within non-motor, there are different types of non-motor symptoms. So just by looking at this complexity of different symptoms of Parkinson's disease patients, you can intuitively guess not all patients will have all of the symptoms. And for the patients that develop different types of symptoms, the severity of each symptoms also differ. This already tells us something just by looking at the symptoms. There are heterogeneous disease subtypes exist for Parkinson's disease. And one of the goals for us, Value Health, is we want to identify those finer scale granular patient subtypes within PD. And then coming down to the lower panel here. So the next intuitive thing for our scientists is why do patients develop different subtypes of PD? And we want to know the root cause. And what's known and believed at the moment in the field is there is a multi-factor type of disease. So there are genetic factors, which hasn't been fully understood as well. There are known risk genes, but we believe there are more to be discovered. And also aging is known to be one of the main factors having patients develop PD, and also environmental factor is another big player here. So those are all the things that we think about from the perspective of the root cause of having PD and the root cause of how those different granular disease subtypes form. So those are all the aims in our mind when we first started conducting this research. So here, what I want to show is actually what we utilize at the very beginning of this project. We utilize the data within Genomics England. There are a very nicely curated HPO term table for those patients, PD patients, within Genomics England. We picked out the top 10 most frequent HPO terms of those patients and did a very classical clustering on them, on those 900 patients. This is only looking into their symptoms. And as you can see and expected, we can see it's clearly those 900 patients can be classified into 4 different subtypes of the PD disease, just based on the symptoms of them. So this is interesting, but also very within our expectation. So the next thing in our mind is why do those patients differentiate into those different subtypes? Because we know the advantage of high quality whole genome sequencing data provided by Genomics England. We want to study this from the genetic perspective on a deeper level. So that the next thing we actually started is to look into their genetic data. Before I go to the details, I want to introduce a little bit more about background on the PRS and loss of function aspect of the genetic data. So as I mentioned previously, we want to study this genome data on different layers. The one layer is the common genetic variant, which we utilize the PRS compute framework. So just to remind everyone, what we have here in Genomics England is very deep whole genome sequencing, on average 30x. And then you can imagine for each individual participant, you will get 4 to 5 million SNPs on average. So that means a data frame with about 4 million columns. At this point, we don't think it's wise to just dump off all of those 4 million columns into our framework. That's why we propose to tease them apart. So what is PRS? It's really a value to estimate an individual's tendency to develop certain phenotype. And to remind everyone again, when we talk about and think about phenotype, the most commonly phenotype we think about is disease risk, but it actually goes a lot more than disease. We can certainly compute a person's disease risk to develop a neurodegenerative disease or Parkinson disease, but we can also compute this PRS on a whole range of biomarker, which is something that we did in this project as well. So if you're interested, you can feel free to take a look of this very classic mathematical equation of how PRS is generated. It actually utilized GWAS study output, GWAS catalogs. The ones that we selected is actually based on public available UK Biobank. So here, what I show is actually what we compute using this Parkinson disease cohort within Genomics England. Remember, it's 900 patients, so we can actually see a nice distribution out of it. So on the left panel here, what we are showing here is the PRS scores of those patients developing neurodegenerative disease risk. So you can see one interesting thing coming up is that you can see this bimodal distribution of those 900 patients developing neurodegenerative disease risk. So that's already telling us something. Even if those 900 patients, they are labeled with one single Parkinson disease diagnosis code, they certainly from their genetic perspective, they can be classified into 2 clusters. And those are the things that we start to look into in our project team as well. On the right panel is what I want to show. Usually, if you compute PRS on the endophenotype, for example, urea level, those kind of common phenotypes, you will expect to see for 900 patients, this is a nicely looking bear-shaped normal distribution. That's usually something we expect. But it's usually when we see on the left panel that for certain disease risk, for the same diagnosis cohort, from genetic data, we see this bimodal distribution that really gets us data scientists excited and want to dig into more. So to switch a little bit to the other spectrum of genetic data, which representing the rare genetic variant. So the loss of function genetic mutation, I'm sure everyone probably have heard about it, and usually this is result in the gene product having less or no function, being partially or wholly inactivated. What type of mutation can cause it? And as you can imagine, stop codon and frame shift mutation certainly will cause this type of impact. It's usually rare and have more severe functional impact. One of the things why we want to study more about this type of genetic variant is because previously the known loss of function mutation risk genes in Parkinson's disease is still very limited. What I show at the bottom of this slide is actually from a public available literature. Since loss of function, variant is so rare, you might think about for a generally healthy humans like us, do we have this type of genetic variant? The answer is yes, we certainly do. On average, a healthy human being might have from 100 to 200 loss of function genetic variant. It's usually about aggregation of those genetic variant, the burden of them, and how those burdens spread it within our genome. Is it spread it like randomly across the 25,000 genes, or is it actually spread it and aggregated onto certain important pathways? Those will have very different symptoms in the end. So those are the things to keep in mind when we start to look into this type of genetic variant. So just to introduce a little bit, so last year within Genomics England high performance compute environment, we leveraged this environment and built up the two different pipelines to tease apart these two layers of genomic data. So one part is polygenic risk score pipeline, and the other part is the loss of function accumulation compute framework. So now switch to introduce how we actually, now that we have those different layers of the genetic data, how can we integrate them? How do we want to integrate them in certain ways? So this is in general a multi-omics field questions. So multi-omics approach has been very popular in the past several years. As I show at the bottom here, this are from Mixomics publication. I like this page because it really represents how we in the field think about what are the different types of omics data. For example, how can we integrate genomic data with transcriptomic and proteomics all the way to other omic data in certain ways that we can generate patient subgroup and actionable insights. And building on top of that, this framework is really not just can be used onto molecular level of data. We can also use it on other data modalities. For example, on clinical phenotypes or imaging or electronic health record type of data. So in this field, there are also different computational algorithms that's built up to solve problems like this. For example, there are matrix factorization and also Bayesian-based framework, and then network-based, and at a very early stage, it's more intuitively you study each layer separately and then find overlap. To think about our questions in our mind for this project is the omics that we are actually projecting is the two layer of genetic data we extracted from whole genome sequencing. One is the PRS representing the common variant layer, and then the other one is the loss of function representing the rare genetic variant. So the method we chose last year when we did this experiment is a unsupervised clustering method called TWL, two-way latent. This unsupervised clustering method is very useful because for our question, we have no prior biological disease subtypes information. So to introduce a little bit about TWL, is a Bayesian two-way latent structure model for integrative genomic clustering. One of the key points of TWL is it actually allows the missingness of samples across different data sets, which will be very useful if you are really into a clinical setting. Sometimes not all patients have all the multi-omic modalities of data. So in our project here, presented here, the goal is integrating PRS and PLOF, and then identify subgroups using those two layers. Once we have the subgroups assignment, we want to understand those subgroups. The way that we do is utilize the HPO disease phenotypes table within Genomics England, finding the association between subgroups identified and the patient phenotypes. Through this process, we want to generate actionable hypotheses and develop experimental plans. So to give everyone a little bit of context about what the output may look like once you have done this analysis. So on the left side here, what I show is actually directly from TWL's publication. So you can imagine, because for our question, we have two types of data modality. One is PRS. You can imagine this heat map A representing the PRS data space. On the x-axis are the alignment of different individual. On the y-axis, you can imagine those will be the clustering subgroup assignment. So it may look like the PRS data space, the 900 patients may naturally fall into 5 or 6 different subgroups. And then onto this heat map B, you can imagine this is a loss of function data space. And then by looking into the loss of function data space, patients actually can only be grouped roughly into 2 large subgroups. So once we have this kind of information, the next intuitive question in our mind is, how can we connect this intercluster assignment relationship? So the way that we did within Valo is we come up with using PV cluster, which is another clustering approach, which on top of giving you the cluster, it also gives you bootstrap values to help you identify the high confidence clusters on these dendrograms. So what I show on the 3 stars here, those are the 3 high confidence interclustering assignments groups we looked into. So LOF represents the cluster assignment of loss of function. PRS represent the cluster assignment from PRS. So you can see from this loss of function cluster 3 matches to PRS cluster 6. Those are properly, you can imagine those may be the same group of patients, but from thinking from their PRS genetic perspective, they are into the PRS cluster 6. But if you look into their loss of function genetic space, they are into the loss of function cluster 3. So now we have these results. What's in our mind is that, how can we understand those clusters? The next thing we did is we, on top of that dendrograms, we added into the HPO terms for those patients. And then what I zoomed in is this part, which also on the top right level here, you can see these are still the loss of function cluster 3, PRS cluster 6, I showed in the previous slide. And this actually also grouped with a cluster of different symptoms. We looked into those symptoms and, for example, favorite response to dopamine medication, resting tremor. And when we get to that point, our group realized one thing. This group of symptoms looks very, very familiar. Remember at the very beginning, before we even look into this genetic data of PD patients, we did a very classical clustering on those patients' symptoms. So I'll show on the next page to remind everyone, remember this page at the very beginning. So those 5 symptoms that's shown in the previous slide, actually, they all come from this cluster 2 of PD patient. So now we are making a lot of connection, linking different pieces together. So the cluster 2 of PD patient, just based on their symptoms, we actually can connect them to different genetic data space. They are connected to PRS cluster 6. They are also connected, associated with loss of function cluster 3 and 5. The next thing is why, or what's so interesting about those genetic cluster. From looking into those genetic cluster, we might can find something interesting to explain why those patients develop this group of symptoms. So I'll show one example in the next slide. So as computational biologists and machine learning data scientists, we always like to start with something very basic and classic. as for model building process. So what we did is we chose to use a group of PRS biomarkers that we compute using Genomics England PD patient data, and added age and gender as covariates to explain this PRS cluster 6. Why are those patients have this cluster 6 PRS assignment based on, so I showed this about 30 biomarker-based PRS, and also we added some disease risk PRS, for example, developing PD, Parkinson disease, or neurodegenerative disease risk. And some of the biomarker we have chosen to include in this model building. I showed 2 publications here. They are already known to be relevant to PD disease. That's why we want to include them. So the model summary stats are not shown here, but if you think about once you have the model there, and then just based on the significant beta coefficients of different features, you can start to look into deeper on why those patients develop certain symptoms for cluster 2 be assigned into PRS cluster 6. So we did a similar process also for loss of function model building. And this is just one step as a baseline. And then on top of that, we can add it into more features to control confounders and also utilize different types of machine learning, not just based on linear regression. We can also introduce different interaction terms. So some about conclusion and future direction. So this project was mainly done in last year. So TWL since last year is not the only method allowing missingness. So we are starting to try new method. And instead of doing genetic omics, identify subgroups, we are thinking about incorporating genetic omics and clinical data feature matrix as one step, and then identify novel clusters and go from there. And besides the HPO terms, we're also looking into other data modalities that's provided by Genomics England. And also, once we have this novel, interesting patient subgroup identified, we want to identify the key genetic marker and the biomarkers. So before we move on to actually pass this hypothesis to our neurology group or to do experimental validation as computational biologists, we always think about how we can computationally doing experiments to rule out false positive results. One example we always like to talk about, because Genomics England have this 100,000 genome project, that's already a very large pool for us to do random draw experiment to perform this type of computational experiments. So the last page is acknowledgement. So first, I want to acknowledge our Value Health internal working group on Genomics England. This is truly a collaborative project. I want to give thanks to a lot of our hardworking, also talented Value Health co-workers. Without them, we couldn't have done so much interesting science utilizing Genomics England last year. And also, if you're interested in joining Value Health, please feel free to check out our company website on the career subpage, and also feel free to check out our LinkedIn and reach out if you're interested. And last but not least, I also want to express our appreciation to Genomics England for making such a wonderful and rich data resource available to scientific community to help patients and find more effective therapeutics. With that, I will stop sharing and turn this back to Parker.
Yeting, thank you so much. What a fascinating presentation. And I think it's really insightful to see that, although we didn't recruit for Parkinson's, that there's still such insight into complex disease in our data set. And it's terrific to see how deeply you've looked into the clusters and the segmentation. We've got a few questions in the chat already, which I'll jump into, but maybe I'll just start off by asking you to just, for a minute or so, just tell us a little bit more about Value Health as a company. How many people do you have? Where are you based? What kind of people do you employ?
Sure. So we Value Health, we actually, the main office is based in Boston, but we do have office across United States at different areas. We have office on the East Coast. As you know, Parker, I'm actually based in New Jersey. We have office at Princeton. In New Jersey, and we also have office on the West Coast in San Francisco. We do have our large group of therapeutics department. We have like a wet bench work biologist, as well as me. I belong to the technology and product department, where we have a group of data scientists and also software engineers. So we are actually actively recruiting for all sorts of different talents. So feel free to reach out and check on our webpage. And we are we see ourselves as non-traditional drug discovery company. And as you see, also introduced, we have our Valo end-to-end compute and machine learning platforms. So we welcome people check us out.
Thank you, Yeting. And we've got a bunch of questions. So some quick fire questions to you, please. First one from me, you mentioned your matrix factorization, and it was mostly genomic and clinical data. Have you included any imaging modalities into your factorization, perhaps more relevant to cancer than in rare diseases?
Yes, that's a very good question. So for this project, we presented today for Parkinson disease, we did not include any imaging data. And this is also my understanding, Genomics England, we haven't actually have actual imaging data available to us, but imaging data is one of the big thing that's going on within Valo on other programs. We are actually looking into imaging data for different programs. With respect to imaging data, we normally would switch our gear to use more like a deep learning approach to do subgroup discovery.
Okay, well, watch this space, everyone. We will be adding digital pathology and digital radiology to our cancer data set this year. We've got a question from Saeed Sayad, which is, how do you deal with the reproducibility issue within genomics data?
Yes, that's very good question. So the way that we do in Valo is for the same Parkinson disease, neurodegenerative disease space, besides Genomics England, we also have access to independent other data sets. So we actually try to reproduce, if we are confident about certain insights, we try to reproduce it in independent data sets and see whether we can find the same signal. And also we are trying to work on different machine learning approach to actually connect different patient subgroups in independent data sets, because they are all controlled data set, and there's no way we can stack patients' data onto the same space. So we're trying to do this in a innovative way.
Excellent. And diversity is an important topic for Genomics England. How was ancestry or ethnicity used, or even relatedness or sex used in your analysis?
Yes. So for Genomics England, and also things that's relevant with UK Biobank, one of the things that we actually feel is a little bit lucky to us is because the ancestry usually will be largely the same. But the way that we usually do is we control ancestry using maybe the top 10 to 20 PCs from their PCA analysis from their genetic data. That's how we usually do in our machine learning approach.
Okay, thank you. And did you find sample size a limitation within our data set, or were there sufficient samples to work with?
That's very good point. So for Parkinson disease, we do feel 900 patients is not big enough, but it is good enough to start to generate hypotheses. That's where I am talking about. We do have independent other larger data sets in different controlled environment. So what we actually leverage on in Genomics England is really this high coverage, high quality whole genome data. We leverage on that, generate insights, and try to see whether that insights can be reproduced in a larger independent data sets.
Okay, but if we gave you a magic wand to request more data or a larger N in any particular area, what would you wish for?
That's really hard to directly answer. I probably can set up a poll in our company and let them to choose which area they want to focus on and ask for more data.
Okay, excellent. I think we have time for one more question. I'm going to go back to a second question from Sayed. I'll just read it out. The majority of data science ML projects are descriptive, not predictive. Have you had any success with predicting, building predictive models, for example, Parkinson's at an early stage, so early detection of Parkinson's?
That's very good point. At this stage, we haven't actually built up like very rigorous predictive model just using Genomics England datasets. Genomics England dataset in our mind is more like a drug discovery type of datasets to go with. But we do use predictive modeling on independent other datasets where we understand that disease better, we have more data modalities, and then that's and the goal will be very straightforward. But for Genomics England, we really do want to do discovery, supervised learning modeling.
Okay, terrific. Yeting, thank you so much for a fascinating presentation. I expect we're going to get a few more questions offline. If there are any participants that want to ask further questions, please send those to me directly and I will get them to Yeting and make sure that answers get back to you. And similarly, as Yeting said earlier in the presentation, Velo Health are keen for academic collaboration in all of their disease areas, not just neurodegeneration, so also heart failure and cancer. So if any of you are interested in participating with Velo Health in that domain, please let us know and we'll connect you offline.
Thank you, Parker.
Thank you, Yeting. Thank you very much. And I'd now love to invite our second speakers to the stage. We have Matt Murray and Patrick Tarpey. Are you guys there?
Yes, we are. So thank you very much for inviting us to talk. I'm just going to share my screen.
Thank you, Matt. I could go through the your lengthy biographies and read them out. Would you like me to introduce you formally, or I'm happy for you to introduce yourselves?
No, I'm happy to introduce ourselves. So I'm Matthew Murray. I'm a paediatric oncologist from Cambridge. I'm split between the laboratory and the clinic 50-50. Patrick?
Yes, I'm Patrick Tarpey. I'm the lead scientist within the East of England Genomic Laboratory Hub, and I play a role in analysing the 100K whole genome data for cancer and the migration of those cancer genome analyses into routine clinical practice, specifically for paediatric cancer at the moment.
Thank you, Patrick. So what I'm going to do this afternoon is just exemplify how whole genome sequencing was helpful in a patient we had presenting through our service in Cambridge, and how it altered our management and how it's actually really helped us to understand a little bit more about these rare tumours, paediatric astroblastoma. So Patrick and myself are presenting on behalf of the whole authorship team, most of whom are in Cambridge, but also Tom Jacques and Jessica Pickles, who are based at the UCLH and Great Ormond Street team, who also provided valuable help and insight into this case. So just in terms of the setup, as part of the Genomics Medicine Centre, we're from the east of England, which is this area here. It's a large geographical area. It actually, the details are shown here, and under the NHS sort of commission service, we're now the East Genomics Laboratory Hub, or East GLH. So how does that map to paediatric cancer? Because paediatric cancer is set up into around 19 different centres within the UK. So of course, these things don't always map perfectly. But in terms of the paediatric cancer, these are the 19 principal treatment centres within the UK and Ireland. And our East GLH, or East of England Genomic Medicine Centre, maps to 3 centres, obviously Cambridge, which is shown here as number 8, and in 7 is the East Midlands Integrated Service, which is based at both Queen's Medical Centre and Nottingham and Leicester Royal Infirmary. So technically our geographical area is covering the East of England, but also part of the East Midlands. So in practice, what does that mean? Most of the cases we did through the pilot phase of the project were Cambridge cases, but more recently we've had input into our molecular MDTs from the teams from Nottingham and Leicester. And in addition, we're a tertiary level service for the whole of the east of England, but for things like bone tumours, which are done at Birmingham and Stanmore, we have particular links with Stanmore, and that's a quaternary level service. So because our patients go there for biopsies, we've also started linking in with Stanmore, and Stanmore linking in with us in terms of contributions to our molecular tumour board. So it just sort of gives you an idea of the level of complexity that's required to make sure our patients have the best access to whole genome sequencing, and that we're accessing, you know, we may be consenting here in Cambridge, but the sample may have been taken, for example, in Stanmore. We're just really establishing those pathways to make sure they're really robust to maximise the opportunities for patients. So in terms of the way we work in Cambridge, we discuss the intracranial cases on Mondays in a weekly tumour board, which is an hour and a half. We discuss the extracranial cases on Wednesdays in a tumour board that lasts for an hour. And then following that, we have the molecular tumour board where we discuss whole genome sequencing results from both intracranial and extracranial cases. That's been well established now for a period of time, and that has got input from radiologists, pathologists, oncologists, genetics team, as well as Patrick and the East GLH sort of scientific team. So going back to the case we originally described, this was a 13-year-old female patient. She presented with a 2-week history of morning headaches, vomiting and visual loss, a very sort of characteristic short history of someone presenting potentially with a brain tumour. There was no prior history of note, and on examination, she was seen by the ophthalmology team, and it was confirmed that she had bilateral papedema or swollen eye discs on examination, and a central visual field loss was confirmed. So as a result of those findings, an MRI head was performed, and this revealed a large 6 by 5.5 by 4.2 centimetre diameter lesion, which was a mixed solid and cystic lesion. And as a result, because of the that had obstructed the flow of cerebrospinal fluid, which flows around the brain and spinal cord, and had caused those symptoms. So she was started on dexamethasone, which is steroids to help reduce and treat the raised intracranial pressure, whilst that was looked into in more detail. So this is an example of an MRI image from the side, which is what we call a sagittal view. And you can see here some of the swelling of the normal fluid-filled sacs, or what we call ventricles, and this large tumour centrally. And this is the solid aspect of it, with a cystic component more anteriorly towards the front. When we look at it from the front, so looking from the front of the patient to the back, we can see that this is the solid area, which is shown here towards the back of the head, and at the front this large cystic area, and that's causing, as you can imagine, some of the obstruction of flow around the brain. And then finally, from above, we get different sequences, T1, T2 weighted sequences, and they give us different information. So again, on this image on the left-hand side, you can see the solid area here towards the back with the cystic area here. And these ventricles, which usually are not very prominent, you can see the black area here on this T2 image is the cerebrospinal fluid. Those ventricles are dilated as a result of the fluid not able to flow properly, causing her symptoms. And on T1, again, on this image, the cerebrospinal fluid, or CSF, is white, and it just confirms this sort of dense fluid, proteinaceous fluid in this cystic area at the front, and this solid area at the back, which isn't all homogeneous. There's areas which take up the dye or the enhancement more prominently than others. So this is a sort of a patient that would regularly present to ourselves and the neurosurgical team who are co-located with us in Cambridge. So following the treatment with steroids, this patient was reviewed through our MDT, and the decision was made for the maximal safe debulking. And in fact, the neurosurgeons were able to get a complete resection. And these are these 3 images from the side, from in front, and from above, which show that the whole of that tumour has been resected. And obviously, there's a little bit of residual areas as an impact of the tumour. So a slightly sort of a dilated area here on the right-hand side, but otherwise a pretty good response. And as I say, these post-op images didn't show any area of disease in this tumour bed or any other disease elsewhere in the brain or spine, which was an excellent result for the patient. So the post-operative recovery was uncomplicated. Dexamethasone was weaned and stopped, and the patient was discharged home pending the pathology results. So the sample was submitted frozen to our pathologist, who looked at it under the microscope. and obviously a lot of pathology is about pattern recognition. And the pathologist reported that this was a very cellular tumour. It appeared to have neuroepithelial type tumour, and they noticed some particular patterns around it. And one of the patterns they noticed was something they describe as pseudo-rosette formation. So around the blood vessels, there's blood vessels sort of shown here and here, the cells form a sort of, there's a particular sort of nesting or clustering of cells in a sort of a ring shape around some of these blood vessels, and that's the pseudo-rosette formation, which is can be seen in Astroblastoma, this rare tumor type. And similarly, the pathology in terms of the special stains that are used, again, is a recognition of a staining pattern which is consistent with different tumor types. In most cases, the sort of pattern recognition is quite straightforward and can take 5 to 7 days. In other cases, the pattern can be quite unusual and take longer, maybe 3 or 4 weeks to get a definitive diagnosis with the benefit of second opinions. And this is one of the difficulties in pathology, is that if it's straightforward, that's fine, but if it's more complicated, you may be thinking about what further adjuvant therapy may be required following surgery. Is any radiotherapy or chemotherapy required? And you can, the earlier we can get the diagnosis, the better. So in terms of the staining, GFAP is a glial fibrillary acid protein, which is seen and is shown here strongly. It's not diffuse, so it's focal in certain areas of the tumor. So that's one feature on immunohistochemical stain. Synaptophysin with another stain, which, as you can see, compared to the GFAP is on the left, is more weakly positive. Again, focal positivity throughout the tumor. And then the right-hand side is a MIB1 stain, which gives us an idea of how many cells at any one time are turning over. You can see that the rate was very low, just a couple of %, and the little brown spots indicate the cells that are active. So overall, this was a tumor with certainly from the appearances under the microscope, which had a relatively low turnover, and it probably maybe it had been there some time before it came to clinical prominence with the headaches and before it obstructed the flow. So taken together, the histological and immunohistochemical features in this case were most consistent with an astroblastoma. So that's something that we've rarely seen through our MDT compared to other sort of glioneuronal tumours, in other tumours such as low-grade gliomas or medulloblastomas or ependymomas. So we looked a little bit more into this tumour type. So relatively recently, these astroblastomas are recognized as distinct tumor entity, and a number of things they're characterized by, which is in particular a rearrangement of a gene called MN1. So the first paper demonstrates that, and another paper published in Brain Tumor Pathology around the sort of 2018-19 time again identified this MN1 rearrangement. And at that time in our pathology department, the MN1 gene alteration, one of the ways you could look by it is by fluorescence in situ hybridization of fish, and that wasn't something that was routinely done in our laboratory. And because this is a rare tumor, there weren't other laboratories who obviously had this set up to confirm the diagnosis. So we're in a position where histologically and immunohistochemically it looked like an astroblastoma. A review of the literature suggested that these tumours are slightly more aggressive in terms of their recurrence risks than other standard low-grade gliomas, for example. So usually following a complete resection, we'd do a scan in 6 months' time. I think we were a little bit concerned because of the potential diagnosis of astroblastoma. And while there were discussions about how we could confirm the diagnosis molecularly, we felt it would be prudent to undertake a more frequent imaging with 3-monthly imaging, and that was initiated whilst discussions were underway and the patient was consented for whole genome sequencing. And in fact, further review of the literature indicates that it isn't just as straightforward as identifying an MN1 alteration in these tumours, and this is a paper published by Wood et al. In Brain Pathology in 2018, and actually looking at multimodal molecular analysis, a lot of astroblastoma cases could actually be reclassified as different molecular entities. And they acknowledge that astroblastoma is a rare and controversial glioma with variable clinical behaviour, which can be indolent but actually can recur quite quickly and aggressively. And they also, more recently, there'd been recognition that genomic profiling had identified a subset of central nervous system tumours with an MN1 gene fusion, which were called CNS high-grade neuroepithelial tumours with MN1 alteration, or CNS HGNet MN1s. And as a result of this sort of area of controversy, they looked at 8 cases in more detail using multimodal molecular analysis. And in those studies, they found that of 4 of their cases that had MN1 alteration confirmed by FISH, 3 of these cases actually grouped with the CNS HGNet MN1 by methylation array. And so it may have actually, rather than being a pure MN1 astroblastoma, may have actually just been an HGNet MN1 tumour. And in addition, 2 of the 8 cases which were clinically indolent and there wasn't a recurrence remained unclassifiable despite multimodal molecular analysis. And their conclusion is that the histology itself may not be specific for any particular entity, including HGNet MN1, and additional genetic characterization should be considered for these tumours. Some contain a methylation profile or genetic alterations that suggest classification of other tumour entities. So I think a lot of information coming out around the time this patient presented that told us that doing MN1 FISH alone probably wasn't sufficient, that methylation may not be informative, and that we shouldn't just be going on the histological classification. So whilst whole genome sequencing was being undertaken, we just started that more sort of close surveillance with 3-monthly MRI, and then went on to do further molecular analyses. The first thing that we did for the tumour was the methylation array, and at this point I'm going to hand over to Patrick to talk us through the molecular side of things.
Thanks, Matt. So yes, this patient's tumour was analysed with the Illumina EPIC array platform and classified using the Heidelberg classifier. So this methylation assay is able to inform the diagnosis of a cancer by comparing the global methylation pattern with equivalent patterns observed in other neurological tumours. So on the next slide, Matt, what we've seen in this case is that the classifier gave a very low calibration score, so too unreliable for an informative classification in this case, despite excellent probe hybridization. On the next slide, Matt, we can confirm this visually in this plot, which compares the methylation profile of this case depicted with the arrow and the grey dot with 6 other similar tumour types. So it doesn't cluster closely with any of these similar tumour types, including the previously classified high-grade neuroepithelial tumours with MN1 rearrangements that Matt's already mentioned, that are indicated in red on this plot. So in summary, these analyses, the methylation array was unable to resolve this case. On the next slide, Matt. So this patient was recruited to the 100,000 Genomes Sequencing Project. And what we have on the left is a bar chart which shows the numbers of mutations identified in this cancer, indicated with the arrow, compared to other CNS tumours. So the bar chart shows different classes of mutations. Blue are small substitutions, yellow are indels, and red are somatic structural variants. So this case was conspicuous in that it had a relatively large number of rearrangements depicted here in red. So what was striking when we look at this in more detail via the Circos plot or the linear copy number plot and structural variant plot on the right is that this excess of rearrangement was concentrated as inter and intra-chromosomal events between just 3 chromosomes, 17, 19, and 22. Otherwise, the cancer was relatively quiescent. The only other copy number change was a loss, an imbalance involving chromosome 9. So this mutation complexity we have constrained to these 3 chromosomes provides opportunity for these genes resident on these chromosomes to become disrupted or rearranged in ways that may be beneficial to the cancer. And one specific class of rearrangement, inversions, constrained to chromosome 22, was responsible for the generation of oncogenic fusion genes, which has been key to understanding the development of cancer in this case. On the next slide, Matt. So what we did in the analysis, when we realized we were investigating a potential astroblastoma, we looked at the whole genome data and we were searching for rearrangements between the MN1 gene and either a BEND2 or CXXC5 genes, which have been previously described in the literature. We didn't find evidence for these rearrangements, but we did find a novel rearrangement involving the MN1 gene, which was obviously therefore quite compelling in this case. This rearrangement was a large 18 megabase inversion, which had breakpoints within intron 1 of the MN1 gene and intron 2 of a novel partner gene called GTSC1. The resultant chimeric gene comprised exon 1 of MM1 fused in frame to exons 3 to 12 of this novel gene, GTSC1. And when we look at this gene, the structure of this chimeric gene, it closely resembles other MM1 fusions involving alternate 3' partners BEN2 or CXXC5. The variant was confirmed by Sanger sequencing indicated in the trace here, and predicts a novel in-frame chimeric protein consistent with the diagnosis of astroblastoma. On the next slide, please, Matt. So we kind of thought we'd resolve this case nice and neatly until we looked at the data in more detail, and we've seen an additional interesting observation in that there was the presence of a second fusion comprising the genes EWSR1 and PATZ1. This was derived again by an inversion, this time 2 megabases again on chromosome 22. And this inversion disrupted the coding sequence of PATZ1 within exon 1 and intron 9 of EWSR1. The position of both of these breakpoints were identical, with an equivalent case in the literature describing an EWSR1 PATZ1 fusion in a novel glioneuronal entity. And the PATZ1 breakpoint, in our case, was in exactly the same codon as that described in the literature. This literature report was also confirmed by RNA sequencing, and as it was identical to what we're seeing in this case, it confirmed the validity of the variant we've identified in this patient. On the next slide, Matt, please. So EWSR1 PATZ1 fusions are known, so they've been previously described in sarcoma, and more latterly, they've been described in a series of neurological tumors, and there's 3 references on this page here. However, this is the first time, to our knowledge, that it's been described alongside an additional fusion, in this case involving the MN1 gene. On the next slide, Matt. So we repeated the methylation profiling 3 years after the original classification was performed. However, as we can see ringed in red here, the calibration score was still too low to enable it to be reliably classified, albeit we ran the classifier this time with additional cancer types to determine if it helped to resolve this case. So on the next slide, we can see this depicted. The index case is again indicated in gray by the black arrow, the small gray dot. And via collaboration with Tom Jacks and Jessica Pickles at GOS, we were able to obtain 3 other brain tumors, gliomas, which similarly had PATZ1 rearrangements. And when we included these in the methylation profiling experiments, these clustered intimately with our case. So what we see is 3 little green dots, which are just underneath the gray dot in this case here. So the PATZ1 rearranged neurological tumors appear to distill into a distinct methylation entity. And to summarize this work, therefore, we have a cancer with 2 distinct rearrangements involving the MN1 gene and the PATZ1 genes, and the methylation profiles cluster with other PATZ1 mutated cancers, but not with other MN1 rearranged cancers. And in the absence of the genome, we wouldn't have conceived that this was a PATZ1 mutated cancer. So what this means with respect to patient management, and the next slide, Matt. So this data confirms a molecular diagnosis of astroblastoma. Astroblastoma confers an elevated risk of recurrence. So the patient had 3 monthly MRI scans rather than the usual 6 months. The patient currently remains well, and all subsequent MRI scans have shown no tumor recurrence over a 36-month post-operative period. on my last slide, just to widen the discussion out a little bit. So what we were able to show in this case was whole genome sequencing providing useful information to inform the diagnosis and the prognosis. And it enabled us to pursue a series of experiments we wouldn't have otherwise considered in the absence of this insight. So what we have on the left here is a graph which shows this case, but also other additional CNS tumours. And what we're showing on the left is a harvest of somatic variants, otherwise unknown via standard of care assays, which provide information to influence the diagnosis, inform the prognosis, or offer novel therapeutic opportunity in other CNS tumours via this whole genome sequencing effort. This information is simplified in the 3 pie charts on the right. So in a cohort of approximately 40 paediatric cancers, WGS provided variants which were able to influence the diagnosis in about 1/3 of cases, inform the prognosis in 8% of cases, and offered therapeutic opportunity in 15% of cases. So as we move from 100K analyses to commissioned genomes, we now routinely sequence pediatric cancers as part of routine clinical service. And this theme of informative, sometimes unexpected clinical insight has already been recapitulated in the analysis of our first 20 cases. So I'm going to pass over back to Matt to wrap up.
So I think this single case sort of exemplifies very nicely as a sort of clinical vignette the benefit of whole genome sequencing. From a tumour characterization perspective, there's good evidence now that just a morphological diagnosis of astroblastoma under the microscope is insufficient to really accurately classify these tumours. Even if we'd had MN1 FISH available to us, this fluorescent in situ hybridization, to confirm the presence of the MN1 gene alteration, which is consistent with an MN1 altered astroblastoma, that wouldn't have been sufficient without further molecular analysis to work out whether it actually was that entity or an HGNet MN1 tumour, or in fact some other type of tumour. We did standard methylation profiling, both at the time the patient presented in beginning of 2018, when we did the whole genome sequencing, but also again late in 2020, almost 3 years later, as Patrick has said, and that was against the DFKZ classifier, which has thousands of patient samples in it and is increasing all the time. So we just repeated that to check that for a second time, how many years later, that global methylation profiling would be insufficient. And I mean, it's important to say that methylation profiling has been really useful for children's CNS tumours, and in about 60 or 70% of cases, it does classify them very accurately with a score of greater than 0.95, which is diagnostic. But you can see that the scores we obtained in this case of 0.04 and the very, very modest improvement to 0.07 is still way off the sort of 0.9 level of confidence that you need to be diagnostic. And in fact, it's the whole genome sequencing results which have proved informative and have allowed accurate classification in this case. And not only that, it has allowed us to identify this PATS one gene fusion that we would have never have known would have existed, and allowed us to instill those 3 cases into the methylation profiling and demonstrate that it clusters with it. So as a result of this, we will learn more over time. We're still not sure as to whether this case represents an MN1 astroblastoma. It certainly does histologically, but whether its behavior might be more like a PATS1 glioma, which have been recently identified, I think time will tell as we learn more. And I think this was an unexpected finding, and hopefully it will contribute to the literature and our understanding over time. But certainly this type of approach will allow us, we were able, once we got confirmation of the MN1 alteration in this patient, we continued that more frequent follow-up, and it allowed us to continue closer surveillance. So if there had been a recurrence, the patient could have had further surgical management before it became problematic and presented with symptoms. So we've published this case now in Neuropathology and Applied Neurobiology. So the PMI ID number is available there for people to look at it in more detail if they'd like. But I'd like to thank Parker Moss once again for this opportunity to present. Thank you.
Thank you very much, Matthew and Patrick. And I must say, I've spent some time with Patrick down in his lab, and the level of care and attention and diligence that you and Matthew pay to each of your patients is absolutely remarkable, and just exemplifies the kind of thing that Genomes England is trying to achieve by providing whole genome sequencing data to the GLHs. So we're very grateful and impressed by what you've achieved here. You've spoken, I think, with great clarity about all of these different modalities and the value that whole genome sequencing adds. But it's not just the additional modality, it's a kind of a new approach of taking a kind of hypothesis-free view on the genome. It slightly speaks to one of the questions from Saeed Sayad, which is, what was your initial hypothesis before starting the experiment? And maybe you could generalize beyond that and just describe how a kind of hypothesis-free approach, how letting the genome kind of tell the story of the patient, you think will contribute to kind of more general cancer care, particularly with kind of complex or refractory or relapsed patients in the future?
I think that's an excellent question. For this case, we sort of had the hypothesis, our hypothesis going into it was that because the pathologist had told us this was consistent with an astroblastoma, our hypothesis was that we would find an MN1 alteration, and we did. In fact, it wasn't the BEN2 or CCX5 standard partners, so it was a novel partner. So we've identified the MN1 GTSE1 fusion, which other people may identify in the future. So I think that was confirmed, but it was novel. So that was the first aspect. The PATS1, so usually tumours have, one main driver. It's unusual to have two drivers. Certainly in sarcoma and other tumours, the second fusion that we found has been found to be a driver. So that was a difficult thing to square, why we'd, how we'd found two drivers. And in fact, it was at the revision stage of this paper that the GOS and UCLH team said, actually, we've just done methylation profiling on 3 PATS1 tumours, so we could add these into the methylation profiling. And so we did that at the revision stage, and actually they clustered with it. So the paper almost evolved in its whole process from first submission to actually final acceptance, in that we had more data over time. And I think this is very much, was the agnostic bit, where we weren't expecting to find anything else, but had to try and fit the additional information that we had into our current understanding and try and actually that was something that evolved over the duration of that submission, which was probably a sort of a 6-month period in total. So I think what that summarizes in just that single case is how our work in this sphere evolves. We may have some hypotheses that we bring from the histology, but actually if we just think that's the only thing we're going to be looking for and we only use targeted approaches, then we're not going to continue to learn. And sometimes it can be difficult to try and juggle this information and try and work out what it means. And sometimes we don't know, all the answers, but by publishing it and putting it out there, we will, over time, will continue to develop and improve our classification of these tumors.
Thank you. And we could see from your images that using the genome browser and looking visually at the genome was a critical part of this. Do you think this kind of artisanal approach that you're taking to every single individual patient will eventually scale and be more automated? Can you see that in the kind of software roadmap of the future?
Patrick?
Yes, I mean, what we're keen to do is to, we're really privileged to have these whole genome sequences available to explore the cancer types in these patients. So what we're really keen to do is to maximize that information. So we really want to utilize as much of that information as possible, and to, so we're in some ways directed in our analysis by the diagnosis or the working diagnosis. So that guides our exploration. But we're very careful to ensure that it doesn't delimit our search for other events. So what we're keen to do is ensure that we look for focal events consistent with the diagnosis, but also explore the genome for any other driver variants which may be contributory or which may inform a slightly different diagnosis, as in this case. I think in this case, it would have been quite easy to have found the MN1 fusion and to feel that the job's complete. That would be entirely consistent with the presentation. So it's ensuring that we don't stop there and we utilize all the available data. So to do that, it involves the resources, the automation and software progression to enable us to clarify the data as much as possible. So I think advances as we increase our understanding, I think the resources that allow us to visualize the data and to triage the data into variants of interest, I think they will develop and mature, and that will, I think, offer opportunity for scientists to do more expeditious analysis and to ensure that the analysis isn't constrained by pragmatic reasons and reporting times, but we really do ensure that we do a thorough analysis in the case. So I think there's opportunities definitely for the software and algorithms we use to contribute a key utility as we progress.
Terrific. Okay, thank you for that. That answered it perfectly. Just a couple of questions from the floor. So here's one from Leo Doherty. Is the MN1 Astroblastoma hiding behind the PATZ1 methylation profile? Is this an isolated case, or may there be other examples?
So I think it's a very good question, and I think that the short answer is we don't exactly know. We haven't got enough whole genome data, and there's probably not enough methylation data with astroblastomas in it. And it also depends how, when the data is submitted, for example, to Heidelberg Classifier, which I think is probably in excess of 10,000 brain tumor cases now. So it is seen as it will classify most tumors, and most tumors we get back have got a score of like 0.98, 0.99, and are very robust. But I think it depends how this was classified. If we classify this as an MN1 astroblastoma, then it may have difficulty clustering. But if we classify it as an MN1 astroblastoma or a PATZ1 glioma, and I think it's almost as if what's driving what, because if we have more whole genome sequences on cases like this, and there is that information available to the classifier, the methylation will undoubtedly improve over time. But but it's not going to get to a point where the methylation will resolve all of these cases. And I think there's certainly cases when you do a methylation and it's unclassifiable, we want to know more information. So I think it's the NHS whole genome is commissioned. How exactly this sits in methylation will partly depend on how some of the cases that have historically been put in before WHO 2016 classification, and there's another one coming out soon, it may need a review of some of those cases. And if they're a little bit historic, or they're called something by an old terminology, you will struggle to get newer entities. It'll take time for newer entities to cluster reliably. So I think there's always going to be that lag phase. Do I think this is genuine in MN1 astroblastoma or a PATZ1 glioma? I think we can't. We've talked about this a lot in our molecular MDTs, where whatever comes up from a molecular perspective, we can't ignore what it actually looks like. So the pathologists who look at the neuropathologists, they're not going to be out of a job. Their role is absolutely fundamental to and it's an integrated MDT where we take everything into consideration when coming up. And often we end up describing tumors as sometimes Wilms tumor-like, or rather than Wilms tumor, because when we look at them under the microscope, they look like renal cell carcinoma, for example. So although the genomics and transcriptomics may look like a Wilms tumor, we can't call them Wilms tumor. So we've got to remember primarily the pathology, but clearly from a molecular perspective, this tumor is behaving in some way more like a PATZ1 tumor on methylation in a small limited data set, but again not classifying yet on the wider DFKZ classifier. So I think all in, what we know is increasing over time, and what we don't know is becoming apparent is even greater, and we'll just continue to go up that curve of knowledge and realize that what we don't over time. But this is a really great way of defining these tumors in more detail, and brain tumors remain something that in terms of overall outcomes have lagged behind some of the extracranial tumors for children. So I think there's certainly this approach will, and the timeliness now of the results coming back within a number of weeks. The patients are recovering from brain tumor surgery, and when you need to make those neoadjuvant decisions, we will have these results available to us. So that's just a real step change in the way we manage these patients.
It's great to hear turnaround time is beginning to work for you. And I think what you're saying is not just MDTs, but multimodality is here to stay for a while. A question from Marzina. Did you check the clonality of the fusions? Is it possible there may be two distinct clones with different fusion drivers?
Do you want me to take that one, Matt?
Yes, please.
I think we did explore this question. So we looked to see what other variants that we had within these data to determine if we could identify the presence of one or more subclones within the data. But the reality was, other than the extreme variability encapsulated within chromosomes 17, 19, and 22, the data was otherwise really, really quiet. So we couldn't really inform whether there was more than one clone in this case. But we can't absolutely exclude the possibility that there's two different clones, each harboring one of these fusions within this case, albeit they would have to render the tissue with the same histological type, because there was no histological distinction within this cancer case. Or it could be that they're resident, both fusions are resident within the same cells within a major clone. But I don't think we had enough data to be able to make that distinction in this case.
No, so I think the assumption is it's the latter, but we do, and I think it's also because histologically it was very homogeneous in terms of its appearance, and it didn't have a biphasic nature to it, which suggested there were maybe two entities, which some of our other tumours within our initial cohort were biphasic, for example.
Okay, good. Well, look, thank you very much, Patrick and Matthew. We had a couple more questions, but I have to say that the ones I haven't addressed are both from internal Genomics England users, so they can get to you at any point, and I'll encourage them to do so. Thank you for answering those questions so well and for that very compelling presentation. I think I'm going to use this opportunity just to close out, as well as thanking our guests. Zan, if you could just pull back up the slide to remind us of what the next seminar is in June. I'd like to present that again. We still have over 100 participants on the call, so that suggests that this has been a successful and interesting presentation for everyone. So thank you for staying until the end. You'll see in the chat there's a SurveyMonkey slide, which is to give feedback on this presentation session and requests for future sessions. Please, can you all respond to that? We look very carefully at it. And then just as a reminder, on the 29th of June at 2 p.m, Dr. Ruth Horn is going to be talking about ethics in genomics, and Dr. Sisodia is going to be presenting about epilepsy. So please do come back for that. It will be Mark Caulfield's final presentation, and he'll be hosting that event. Thank you to everyone for attending.